Hello everyone, my name is Greg Clark and I'm here talking with Scooter Cheatham of the Useful Wild Plants Center today. It's September 2nd, 2022, and I'm going to start off by telling you my story, how I got interested in plants. And when I was a high school student, my favorite class as a senior was a biology class, mainly because the teacher was very engaging. And so when I came to Austin, to UT Austin, kind of by default, I was a pre-med major. And I was doing really well in all of my classes, making A's. I was really, as a student, I was very good at studying for exams. I could study the night before an exam and come in the next day and get an A. And so I was getting all A's, and I think it was my sophomore year, just happened to take an elective, plant anatomy with Dr. Jim Mouseth. And Dr. Mouseth is one of the top plant anatomists in the world. And so I took this class with him, and it was the first class that I took at UT where there was undergraduate and graduate students. And so I was enjoying the class and figured I was going to make an A in this class just like any of my other classes I had taken. And I came to his first test, and there were 2 essay questions on the test. It was a much different test style than I was used to, which was multiple choice, short answer. And I failed the test miserably. And I went into his office and talked to him, and it was really a realization for me at that point that if I looked back at the first couple of years of classes I was taking at UT, I was getting great grades, but I wasn't actually learning as much as I should have been. And I ended up, turning the ship right in that class. I passed the class. I did not get an A, but I learned more in that plant anatomy class than I had any of my other classes. And I think the very next semester I took a plant physiology course with Dr. Stan Rue. His style was to teach from the published literature. He didn't have a textbook, so I was really learning about science and how dynamic science was and about how discoveries were made in plant physiology. And I started working in his lab as an undergraduate. He paired me with graduate students and postdocs that were doing research in his lab, and I fell in love with doing research. I had played sports when I was growing up, and I always liked competition, and to me, doing an experiment was like a competition. The first experiments I was working on was purification of this photoreceptor protein called phytochrome. And I would ride my bike into the lab at 5 in the morning. I was the first one to arrive there and would set everything up for the experiment. And the experiment would take 18 hours. And so we were in a dark room with just green lights and cold rooms, and we had to do every step correctly so that at the end of the 18 hours we would end up with purified phytochrome. And I just loved the challenge of that, and fell in love with doing research. I became a scientist because of the thrill of discovery. I can go months in the lab with things not working so well, and that's typical in science. But as long as every once in a while I get to see data that no one else on the planet's ever seen before, that's very exciting. You get to decide what it means. You get to decide what the next experiment should be. So I ended up switching out of pre-med to botany, and getting my undergraduate degree and doing research with Stan Rue in his lab. And this was the days when botany was one of the top departments at UT Austin. It had several National Academy members in the faculty that were teaching and doing research, and it brought in more money to the university than almost any of the other departments. And so I did my PhD with Stan. I had a very exciting project. It was kind of a high-risk project. The kind of research that goes on in our lab is looking at early signaling steps. So plants, you may not have thought about it, but wherever a seed germinates, the plant is stuck there. And so they can't just walk away on a cold day, and instead they need to sense changes in their environment, like changes in temperature, and then respond to those in order to survive and continue growing and developing. And so plants have evolved exquisite ways of of sensing changes in their environment and responding appropriately. And the research in our lab focuses on the early signaling steps. These are the steps that happen like that. And they determine the downstream signaling steps because it's a sequence of causally related steps. And so my research over the last 3 decades have been focused on making those kinds of discoveries. So specifically what I'm doing these days in the lab is looking at a kind of novel signal in plants called extracellular ATP. So even students that I do outreach with in middle schools, they know that ATP is the energy currency of the cell. Inside the cell it drives all the metabolic reactions. But what students don't know, even graduate students sometimes, is that outside of the cell, ATP, as extracellular ATP, acts as a hormone-like signal. And it's been well studied in animal cells, been known for over 50 years that it's an important signal. But in plants, it's a fairly recent discovery that it can regulate growth. And if you're a plant, growth is pretty important. So we've been doing experiments kind of to establish and determine, is extracellular ATP as important a signal as we think it is. Yeah, so that's kind of a brief introduction to myself, how I got into plant biology, and where I am today in my research. The topic of GMOs, genetically modified organisms, is a really controversial topic. You know, if you go to the internet and look for information on GMOs, you're likely to go to websites that are either, you know, very pro-GMO or very anti-GMO. And you know, if you look at Europe, for example, the sentiment there is more anti-GMO, you know, than it is pro-GMO. U.S. is a little bit more accepting of genetically modified organisms. So from my perspective as a scientist, I kind of break them up. You know, there's a model plant called Arabidopsis that plant scientists study all over the world. As a wild plant, you know, it doesn't grow here in Texas. It's too darn hot here. But it grows in the northern U.S. and Canada. It grows throughout Europe and some places in Asia. But Arabidopsis was the first plant to have its genome fully sequenced. And it's kind of an interesting story because you know, the sequence of Arabidopsis came out a year or so before the human genome sequence was released. And one of the first things that a scientist can do when they have the full sequence of the DNA from an organism is they can use algorithms to predict how many genes are there. And so when scientists did that with the Arabidopsis genome, they found, depending on the algorithm, you know, something like 20 to 25,000 genes were present in this little plant. It's a weed, by the way. You know, you can't eat Arabidopsis. It's in the mustard family. But it's a model system for research because it's simple and we do have the full sequence of the genome. But it's interesting, you know, I'm a plant biologist, so I like to brag about plants. When the human genome sequence was released, scientists could use those same algorithms to predict how many genes were there. And of course, biologists thought that, you know, we think pretty highly of ourselves, that, you know, we would have a lot more genes than this little old weed. And it turns out that when they did the analysis of the human genome sequence, there was about 20 to 25,000 genes. And oftentimes you can even see pair up, you know, an Arabidopsis gene with a human gene, you know, and see that they're related. So anyways, GMOs, you know, the first kind of category of GMOs I think of is GMOs that are used for research to answer a question. So one of the cool things about Arabidopsis as a model plant is that it's really easy to get a loss of function mutant of Arabidopsis. And what I mean, a lot of scientists will call a loss of function a knockout. You've knocked out one of those 25,000 genes. Now it cannot make the protein that it codes for. And it's a very powerful tool in research because, you know, a lot of genes we still don't know the function of. And you can get an idea what a gene might, the protein of a gene might do in the plant by studying a plant that can't make that protein. So I can go to my computer and order a knockout in Arabidopsis of any of the 25,000 genes, and if I want to splurge and use FedEx, I can have that knockout mutant delivered to my lab in a week or two. And then I can do experiments to try and understand what that gene does. So that would be an example of a genetically modified organism, a GMO. It's not meant for anything except basic research to understand the function of a gene. If you compare that to, say, what a medical scientist would have to do, the model for them is mice. To make a knockout mouse takes over a year and thousands of dollars. So I'm way ahead of the game, as far as learning about gene function using the model system of Arabidopsis. But when people think of GMOs, they're thinking more of GMOs that have commercial value and that are out there in nature. And so from my perspective, the first GMO in plants was GMO papaya. So there's a wonderful story of Dennis Gonçalves, scientist at Cornell who was from Hawaii. The virus that was attacking the papaya in the Hawaiian Islands was the ring spot virus, and he came up with the idea to make a genetically modified version of papaya that would be resistant to this virus and save the papaya industry in Hawaii. And so he took a coat protein from the ring spot virus and transformed papaya with it, and long story short, he saved the papaya industry. I mean, there's the rainbow papaya, I think, was the first one that he had made. Interesting story in the sense that he had, if you think about Hawaii, you think about nature and beauty, and the people living there were not, welcoming to this idea of having a GMO papaya. And so a lot of his job, what he had to do as a scientist, was convince the papaya farmers and convince the Hawaiian people that this was a good thing and that it wasn't going to destroy their beautiful environment and ecosystem. And he succeeded at that. I think there's a movie that just came out this past summer, I haven't seen it yet, called Food Evolution, which tells the story of the GMO papaya. But there are legitimate concerns about the safety of GMOs, and there's been some commissions that have really looked into this question of GMOs and what they might do in the environment that could be negative. And largely what these commissions found is that so far there's no evidence that it's doing bad things in our environment. But I think scientists have to stay vigilant. One of the concerns of GMOs is that, big ag companies like Monsanto, I think if I even say the word Monsanto, most people have a negative reaction. And that is a concern that, the big ag companies who push these GMOs and can make money off of these GMOs, well, their bottom line is making money for their investors. And so maybe we shouldn't be trusting them with such an important aspect of our environment. So, I think my position is that everyone should look at evidence and decide how they feel about GMOs, but genetically modified organisms, especially with this new innovation called CRISPR that most of you have probably heard of, is a way of doing faster breeding for agriculture and addressing some of the concerns that are probably coming up with climate change, making drought resistant and pest resistant and pathogen resistant crops. It's more or less like breeding. The story of teosinte and how quickly it became the corn that we depend on as a food source. This genetically modifying crops can do it even faster. And CRISPR is a way that makes it more accessible to scientists to make those changes. And an interesting difference between CRISPR and genetically modified organisms is that CRISPR modifies the genome, the existing genome of say a soybean. Whereas GMO, sometimes you can take a gene, say like the Bt gene from insects, and put that in plants. That's something that's not going to happen in natural evolution. But modifying genes that already exist in soybean, say using this new method CRISPR, that's something that could happen during evolution. And so it's a little bit of a different animal when you're thinking about, you know, the dangers as well of genetically modifying organisms. So in my research, you know, I do very basic research on cell signaling, and you know, it's almost a surprise to me that any of my basic research might have applications that could help people in the world. But it turns out that, you know, this extracellular ATP signaling pathway that I've studied in plants, that it does regulate growth, and there are these enzymes that play a major role in this signaling pathway because, you know, whenever you turn on a signaling pathway, say with a hormone, you need to be able to turn it off. So just like the signaling pathways that extracellular ATP turns on, they need to be able to be turned off. And there's this family of enzymes called apyrases that have really high specificity and high activity toward ATP and ADP. And so when they're functioning in the extracellular matrix, they function to turn off the signaling pathway that extracellular ATP turns on. And so we have, you know, started out, you know, just using loss of function mutants in Arabidopsis and studying them. You can also create gain of function mutants that make more of a particular gene product to learn more about the function of that gene. And once we found that these apyrases, maybe through regulation of extracellular ATP, can regulate growth, that we decided that we might try the proof of principle in a commercially valuable crop like soybean. And what we found was that when we express a PA pyrase in soybean plants, we get higher yields and we get increased drought resistance. And we published a paper on this about 3 or 4 years ago now, and our research is funded by a local biotech company called Texas Crop Science, and we've continued with that research, and those soybeans that are modified to express this PA pyrase are GMOs. And so, you know, again, from my perspective, if you can make soybean plants that are drought resistant and that have a higher yield, this is a good thing for people, because as our population increases and we have to grow crops in less desirable places on the planet, you know, we're already growing crops in all the best places, right? But as we need more and more food production, you know, and the climate is changing, you know, having crops that can handle more drought would be a good thing, and having crops that have an increased yield would be a good thing. And so I'm excited that my research might lead to that improvement. Scooter's asked me to talk a little bit about the old days with the botany department and what the current situation is with the plant biology program at UT now. And so when I first joined the botany department, it was either the top botany department in the country or second. It would rotate between UC Davis and UT Austin, who was the top botany department. And it was multidisciplinary. There were lots of scientists studying plant taxonomy, and there were cell biologists making discoveries about plants, and physiologists studying plants like Stan Rue. And so in those days, I think that actually the research was more interdisciplinary. Even if you were just collaborating with another scientist in the botany department, that was a more common occurrence. And we had, like when I started graduate school at UT Austin Botany Department, I don't remember the exact number, but I'm going to guess that there were probably another 20 graduate students that started the same year that I did. And Now, unfortunately, the plant biology department at UT has become much smaller, and it certainly, not having as many scientists doing different types of things, that cuts down on productive collaborations, interdisciplinary um, research, and I think generally speaking that's a problem in science, in all different fields of research. It's very rare where you get collaborations between a scientist in one department and a scientist in another department. Stan Rue and I did that about 10 years ago, where we had a project and we're getting some exciting results, and realized that if we were to collaborate with a professor in the chemistry department, that we could really get some more clear answers to the research questions we were asking. And that turned out to be a very successful collaboration that resulted in a key publication for us. Um, but I think more of that needs to happen generally in science, and certainly in plant biology as well. One of the things that Stan and I have worked on is getting funding for plant biology. I believe Stan Rue and Malcolm Brown started this Plant Research Institute at UT and started getting donations toward it. And the hopes would be that if we can get enough money in this fund, that then invest it, and then the return from the investments could help fund graduate students in plant biology. Right now at UT Austin, there's just a handful of plant biology graduate students, and it would be great to maintain that and even grow that and have more students studying plant biology. Even in the undergraduate program at UT Austin right now, there's only a handful of plant biology students. And in the long run, I think that's something that needs to change. I believe personally that it's really important to learn more about plants and study plants, and to do that we need more students. When I was a graduate student and a postdoc, I always thought that my big contribution was going to be the discoveries I made in my research. And I've published over 50 papers and been really lucky in my research and made some great discoveries. But at some point, I realized that actually the more important thing that I could do is to support students doing research in the lab with me, and support students interested in plant biology research in my classes, and try to grow the interest in students in having a career in plant biology research. And so that's actually become really what I think I can do to help in the long run. Will make much more important impact than the discoveries I've made in the lab. And so I've gotten involved in doing outreach. It was almost accidental. I'm part of the Freshman Research Initiative at UT, and I was one of the first streams to do research. The research that my students do has to do with root hair growth. And so, if you look at a root, there's the skin of the root, and out of the skin of the root are these single cells that are called root hairs, and they greatly increase the absorption area of a root. So they're critically important in absorption of water and nutrients. And if we can understand how to grow more root hairs and how to grow longer root hairs, that's something that could help in agriculture. And so my freshman students study this question and do experiments in the lab on this. But when I joined the FRI, everybody who was teaching in the FRI had to pick some outside component. You couldn't just teach students how to do research and work with the students. And it was just a twist of luck that I became the outreach director for the FRI. And so I started going to elementary schools and middle schools and high schools around town, and started some programs to do outreach to these students. I really became convinced that middle school students were the perfect age to do outreach with, and I started a middle school program called Shadow a Scientist. It's a free program where middle school students come to UT in the summer. They get paired with a scientist, get to visit that lab of the scientists, and get to see that scientists are just normal people who decided to have a career in science, get to see the equipment that is used in their research and learn about their research. And so I've been doing that program now for 12 or 13 years, and it's a very rewarding program. Every once in a while I say, why am I doing this? But then when I see the kids come back from the labs and they're super excited, showing me a gel they ran in the lab or something, I'm reminded that it's well worth my time and effort to do this outreach program. Another outreach program that I've been involved with for almost the same length of time is called Present Your PhD to a 12-year-old. And in this program, I go to middle schools and I present my PhD research. I dust off my thesis and share with the students what a PhD is. Why would you want to get a PhD? And then share with them the research I did as a graduate student. And of course I did research on plants, so I get to tell the students why I think plants are so awesome and expose them to some plant research. And then I take graduate students from UT and they go and practice their science communication skills, telling the middle school students about their research. And the middle school students, they play a key role too, because they grade me and the graduate students' presentations. We have a rubric, and they love grading adults, But they give us feedback, which helps us to become better science communicators. So when I use that term science communication, what I'm talking about is the ability of scientists to communicate research or science to the public, to non-scientists. And I've been involved in teaching an online course where other scientists come to me to learn how to communicate science. One of the keys is paring down the amount of scientific jargon. If you try to talk to a non-scientist and you keep all of those scientific jargon, you're speaking a different language, and you end up losing them. You don't engage them in it. But scientists can learn how to communicate to the public, and I've been teaching an online course for the American Society of Biochemistry and Molecular Biology, teaching other scientists how to do better at that. One of the things that maybe has started this revolution in having scientists do more science communication is that about a decade ago the National Science Foundation added a section to grant proposals called the Broader Impacts, where a scientist who is making a grant proposal to get funding to do research in their lab need to indicate in their grant proposal how this is going to have a broader impact on the community and the public. And so that started to encourage scientists getting more involved in science communication. And I strongly believe that part of being a scientist is communicating science and research to the public. It's not okay just to do your experiments and go to conferences and talk to other scientists about your research. If your research is important, then you need to be able to articulate why it's important to the public. And we have a situation here in our country where there's a lot of anti-science sentiment, and a lot of scientists will say, well, that's not my problem. But I actually think differently. I think it's up to scientists to become better at communicating the importance of science and the basics of science to the public, and that will hopefully lead to us having a more science literate public. And so getting students involved in plant biology and in science is one of the goals that I have. And I've been fortunate enough to be able to do some outreach, and I really enjoy it. A lot of times the scientists are kind of hesitant at first to get involved in outreach, and they want to know what's in it for them. And I always tell them that if you can explain your research to a 12-year-old or to an 8-year-old even, or to anybody in the public who's a non-scientist, then you're going to be much better at explaining it to other scientists at conferences where you go to present your research. And the other thing that I get from doing outreach is, I go to these middle schools and even elementary schools, and the students at the elementary schools say, well, hey, Dr. Clark, you did that experiment wrong. You should have done this or that, They're very curious about science and how science is done. One of the reasons I became a scientist is I'm a very curious person, and curiosity is one of those things you really need to be a successful scientist. So I think when scientists go out and communicate their research to the public, one of the things that they can also benefit with is a reminder that curiosity is what drives science, and probably a reason they became interested in science. And sometimes if you just stay in your lab and just talking to other scientists, you lose sight of the big picture, the curiosity part. That's one of the joys of being a scientist, is asking questions and being curious about the natural world. Yeah, I hope to get more and more scientists involved in outreach, and certainly in plant biology, the Useful Wild Plant Center, that's one of their goals, is getting more and more people aware of this, the great uses of natural plants and the importance of plants. There's a term that plant biologists have come up with called plant blindness, and really what that means is that the public, someone off the street, actually doesn't really think about how important plants are, and they don't even see plants. There's been studies that have shown we're much more liable to see a cute puppy and give our attention to that than rather see a beautiful tree and give our attention to that. And we lose sight of the value of plants in society. And the way I got connected with the Useful Wild Plants is this course at UT called the Hidden Treasures of Plants. And this course is much different than any other course I've ever taught, because I'm a plant physiologist, a molecular biologist, biochemist. This course talks about the value of plants to our planet and to society, and it's such a joy to teach it. The students that are in the class are first semester freshmen. It's a signature course that's meant to build community and help the students learn more about the resources at UT and make that transition from being high school students to being scholars at the university. And they get to pick plants that they want to do research on, and then do presentations on those and write papers on those plants. And so they start to learn about the value of plants. My only thing is, I wish that it wasn't just 18 out of the 50,000 UT students that were learning about plants each semester, that more of the UT students were exposed to that. And I know the Useful Wild Plant Center, that's one of their goals, is education and getting the public to become more aware of the value of plants, and I fully support that. It's a hard nut to crack, though, because everyone who takes a course at the Useful Wild Plants, or almost everyone, loves it. Everyone who takes my Hidden Treasures course love it, but we're only getting a sliver of the audience that we need to learn more about plants. So I think outreach by plant biologists, outreach by the Useful Wild Plant Center, those are avenues which we can get more people involved, but it's a big challenge. As I indicated, I became more aware of the Useful Wild Plant Center through this course, the Hidden Treasures course. My students come and visit the Useful Wild Plant Center and learn about the great work that is going on there, categorizing wild plants. And currently there are 4 volumes, and when I say volumes, I mean these are thick, and they're full of information that no one else in the world knows about the useful uses of wild plants. And so part of the Useful Wild Plant and the EarthFit project is to look at how are we going to save our planet, how are we going to teach people the importance of plants, and we need to do it using educational outreach, using this information that has already been collected and making it more accessible. So, you know, if you're a scientist and you want to work on a particular species of plant because it has this benefit, you can learn about the benefits, learn where you can find it, and start doing experiments. You know, I mentioned earlier CRISPR as a way of modifying plants, and CRISPR definitely will make it easier to make changes in a particular species of wild plant, and maybe make it even more useful. So those are some of the goals that the Useful Wild Plant Center and the EarthFit project have, and it really comes down to having the funding to do and to continue adding. I mean, if there's a chart on the wall behind me that shows where we are in cataloging and learning and writing about these different species that are useful, and we need more funding in order to continue that project. And so to be successful at this endeavor, it's going to take a lot of effort on a lot of different fronts, and in almost every case we need funding for that, and that funding can come from many different sources. But you know, I would say if you're interested in plants, you know, and you think that you can help, that would be a start. And then as we get momentum and making it more accessible to scientists and scholars and students around the world, hopefully people will begin to realize how important this project is. And once people start to realize how important it is and how unique it is, it's not like there's a Useful Wild Plant Center, you know, in every state or in every country around the world. There isn't. There should be more of them, and maybe there will be. Maybe this will be an example that will pop up in other places. But right now, unfortunately, it's a unique endeavor, and it's one that needs all of our support.